This is the prep recording for lecture 6. There might be 2, but I will try to get through it efficiently so that there's only 1. So this is between the lecture about how the genetic code was determined, how it was decoded, if you like. And then the next lecture will be about the steps of translation, which are actually shown here, the elongation stage. But this one will just be looking at different kinds of mutations. I anticipate that maybe lecture 5 will spill over a little bit into lecture 6. And so this might be a little bit shorter than a typical class would be. Okay, so these are the learning outcomes. This one was probably covered in the previous lecture, and the same with this. So from lecture 5, this information, that should have been underlined. So be able to explain what it means, that the genetic code is almost universal, that it's degenerate, what does that refer to, that it's unambiguous. And it would be helpful to know the start and stop codons, especially as practice for some of the kinds of questions you might be asked to answer. For today's topic, we want you to be able to distinguish the germline and the somatic mutations, distinguish these types of mutations. So these are the main categories. In our class, we'll be focusing for the most part on the germline ones. These are the ones that are about actual heredity. And of these mutations, we're interested in frame shift, point mutations, and then different categories of point mutation: silent, missense, and nonsense. So we'll go through all of these, and I have one example of a missense mutation. And then the idea is that if I was to be able, and we'll go through this in the lecture, if I was to give you DNA strands and a protein sequence, that you would be able to look, work backwards and tell me which of the DNA strands was the template strand or the coding strand. And given DNA strands and being told what the template and coding strands were, be able to say what the protein sequence would be. And this is important for in vivo, especially because you will have to find a start codon. Oh, that's vivo, but not for in vitro. So in vitro work, which is the work we really described before when we were talking about how they figured out how the genetic code worked, that was done in vitro, and they didn't rely on start signals and that sort of thing. So at the end, I'll talk about the difference between these and why it's important to know which it is before answering the question. And then given a wild-type DNA sequence and then some kind of mutation is described, be able to predict the effect on the resulting protein. So you'll know the wild-type sequence, you'll know the protein sequence, and then what would happen if this A was changed into a G, or what happened if we deleted this G and this A, and so on. And then you'd have to write out the RNA sequence and determine what the amino acid sequence of the protein was. Okay, and this is the conceptual framework. We're still here, and we're talking specifically about mutations here. So, and I'm just going to skip this because I did cover it in the previous prep, and we will have covered it most likely in the previous lecture. And if not, we will talk about it in class. Okay, so the categories of mutations. There's 2 broad categories, and one is referred to as somatic mutation. So the soma is the body of you, or of any organism. And making mutations in, the cells of your liver can lead to some phenotype. Maybe one of the enzymes isn't working so well anymore, and you have some symptoms or something like that. Or maybe in your eyes, there is a tumor that develops due to a mutation. These are called somatic because they occur in your body. But if you have a mutation in some cells of your eye, those are not going to be passed on to your offspring. So they're not hereditary. They are of interest, though, because They are of interest, though, because those of you who are interested in medical things, cancers are basically genetic diseases, so they're caused by um, somatic mutations. Sometimes there are inherited mutations that will predispose an individual to a cancer, but still other mutations have to occur in order for that cancer to develop. So there's medical interest here, but these phenotypes can't be passed on to offspring. So just think about if you go out into the sun and you get a suntan, if you have a child, that child's skin isn't going to be darker because you had a suntan, or they're not going to be more muscular because you worked out and built up, really good arm muscles or something like that. So that's the difference between somatic and then germline mutations, which are the ones we're interested in. So these occur in gametes, or the cells giving rise to these. So they could be pre-existing mutations that an individual has, but at some point that mutation arose in either an egg or a sperm, or an ova or a sperm, depending on what the organism is, or the cells that give rise to these. And these are referred to as germline because these are the hereditary ones. So this is where our course is focused, because we're interested in the passing on of those mutations to offspring. And this is just an image, and you can look at and read this for yourself. I've already explained this, but it just basically shows non-heritable versus heritable. Heritable can be inherited by offspring. Non-heritable does not get passed to offspring. Okay, so I want to go through a quick, this is the kind of question that you could be asked. And so here's a DNA sequence. The 5' and 3' are labeled, and that's really important, very important. And then if you know that this is the coding strand, then you know what the messenger RNA sequence is going to be, because it's going to be the exact same sequence as the coding strand, except all of the T's are going to be replaced by U's, right? And the RNA is single-stranded, whereas the DNA is double-stranded, but the 5' and 3' are the same. And so if we were taking this this sequence, I would say, here's some DNA. This top strand is the coding strand, the bottom strand is the template strand. So tell me what the RNA sequence will be, and this is how you would do it. You'd look at the coding strand and just replace the T's with U's. The rest of it's the same, and the 5', 3' is the same. And then you look for the first AUG. That sets the reading frame, because AUG is the start codon. That's where translation begins. And that's going to be methionine. And this is one that you should remember. And then the next one is CCA, which just because of something else, I happen to know this is proline. And then the next one is GGC, so that's glycine. And then GH, I can't remember what the rest of these are, but you've got some additional codons here, and then UGA is one of the stop codons. And again, it's helpful, it'll save you time answering questions if you remember the start and the stop codons, because then you only have to look up the other ones. So this is the idea. Tell me what the amino acid sequence would be, and this is what it would be: methionine, proline, glycine, glutamate, isoleucine, leucine, tyrosine, glycine, and then you put the C on to indicate that that's the end of the protein, right? And you label the N terminus. So when you have 5' and 3' for the coding sequence, they are going to 5' matches the N terminus of the protein, and 3' matches the C terminus, and that's the collinearity that we talked about in a previous lecture. Okay, so what types of mutations? We already talked a lot about kinds of frame shifts when we talked about the messing up of the reading frame, and the evidence that DNA was a triplet code was read in threes. Because if you add 1 or 2 nucleotides, or delete 1 or 2 nucleotides, then you don't have the proper groups of 3 anymore. So I won't spend a huge amount of time talking about this, except that those are what we call frame shift mutations, because you have the correct frame shift, but then, you know, there's like a nucleotide missing, so it spills over to the next nucleotide and you end up getting the wrong information. So remember, the fat cat ate the big rat, or whatever that statement was. If you're missing the one of the letters, it'll be the fat cat, and then it would be ATT, HEB, and so on, and these would all be the wrong letters, so they wouldn't make any sense. They wouldn't have the correct amino acid sequence. Now, just to be aware that there are mutations called indel type mutations, insertion and deletion type mutations, and they don't have to just be 1 or 2 nucleotides. You can have little chunks of, you know, 10 nucleotides removed or inserted when there are mistakes in replication, as well as in crossing over and things like that. We'll talk about crossing over later. And so the point being, though, that the amino acids after the insertion or deletion point are going to be incorrect. And so what that means is, if you make a frame shift right at the end of a gene, and like maybe the last 3 amino acids are wrong, probably it's not going to affect the protein sequence. But especially, you know, in the first half of the gene, a frame shift is going to be disastrous, because only the small N-terminal amino acids will be correct, and everything after that will be wrong, right? So these can be incredibly serious, and they tend to be what we call complete loss of function mutations. And I will talk maybe at the end about loss and gain of function. Now, point mutations are different. In point mutations, most of the time, it's a single nucleotide that's altered to another. And this can happen for a number of reasons. Mistakes during replication. Sometimes the wrong nucleotide is put in by the DNA polymerase. And then exposure to chemicals, exposure to radiation, and so on, that cause damage. And then when that damage is repaired, the repair can either return to the correct nucleotide or sometimes leave in an incorrect nucleotide. So you have amazing DNA repair capabilities in your body, and so do pretty much all other organisms. So most of the mistakes that are made in replication and exposure to chemicals, the repair of that and so on, those are mostly fixed, but a few persist. And those of you who are most interested in more ecological, environmental, sort of evolutionary kinds of questions, if there weren't occasional mistakes made, we'd never have had any evolution or any change of organisms, right? Because it's mutations that occur just randomly, and every once in a while one of them causes a specific phenotypic change that actually makes a little bit of an advantage in survival and reproduction, and then that leads to, you know, the alteration of organisms and their adaptation to the environment. Not that it's a goal-directed process. It is, the mutation part is random, but then whether it benefits you or harms you, that's the sort of the natural selection part. Okay, so types of point mutations. The first type is silent, and you should basically fill in the correct word here. This will help you test whether you understand some of the terms that we covered last time. But some kinds of DNA changes don't actually change the protein product. The gene is different, the DNA, the mRNA is different, but then the protein is exactly the same. And the example is, you take some of the codons where you have 4 different codons that, or sometimes more, but in this case, I'm thinking sort of proline, CCA, CCC, CCG, or CCU. Are all specifying the proline amino acid. So if you had CCA in your gene and somehow there was a mutation and you turned that into a CCG, yes, that's a genetic mutation, but no, there's no consequence because proline is still going to be put in to your protein when translation occurs. And so this is the example. Here's our start codon, the next one, proline, the next one is glycine, and GGC is what was in the original. So suppose it was changed, so there was a mutation made, and now we have an AT pair here instead of a CG pair. But when we go through the translation process, GGA gives us glycine just as GGC did. And so we put the same amino acid and everything is perfectly normal. So you can go back to that previous slide and you can see that the amino acid sequence of this gene is, of this protein is exactly the same. Okay, so that's what we mean when we call it a silent mutation. It's a mutation, but there's no sort of consequence to it. There's no effect of it. It hasn't changed anything. So it doesn't count in a way because it hasn't made any difference to the organism. Okay, next is missense. Missense means the wrong sense or the wrong amino acid. So what this does is the nucleotide change actually does change the codons, and so it changes, a codon for glycine into one for glutamine or something like that, or glutamate. And so these can have a huge or a minor effect. A lot of times a small change in one amino acid in a big protein, think of a protein that folds in a certain way, and it's got a binding pocket, it's an enzyme, and the substrate binds here. Maybe there's an amino acid change here, and in fact it's not very consequential. It doesn't necessarily hugely change the protein's function. It may not make any difference to the function, or it may slightly alter it. But if the amino acid changes in a very consequential, very important part of the protein for its function, you could have a serious effect. And so I'm going to show an example of a serious effect, but just remember that a single nucleotide change that leads to a single amino acid change may or may not have a very strong functional consequence, and often it's a pretty weak effect, so it's not a particularly strong effect. And so here's an example. This is our same GGC from the original, but suppose now instead of the C changing, the A changes. Now AGC encodes a different amino acid, and so now we have glycine has changed to serine. Because this is a point mutation, though, it's only affecting one of the amino acids, and all the other amino acids to either side of this mutation are exactly the same. Okay, so it often has a really minor impact, but there are some known mutations that have very serious impacts, and those of you, of course, who are interested in medicine or medical topics will find that of interest. So for example, cystic fibrosis is a very serious disease, and many of the mutations that are identified that that cause this condition are single nucleotide changes. And I introduced some time back a talk about sickle cell anemia. I think it was in lecture 2. And there are several types of mutations that contribute to sickle cell anemia, but there is one that is a missense mutation. And so this just shows you, this isn't the whole gene or the whole protein, of course, this is just part of it. But right up near the front end of the protein, remember, for the DNA, this is going to be the 5', and then for the protein, that's going to be the N terminus. And the mutant is just a single nucleotide change, an A to a T, and it changes the glutamate to valine. And these amino acids are different enough that the mutant sickle cell hemoglobin completely doesn't fold correctly. And just notice all the rest of the amino acids are the same. So this is an example of a very consequential mutation. this is, I'm not expecting you to remember this or memorize it. It's just these different, the blue and the red strands are normal hemoglobin. They form this kind of compact structure that you almost, when I think about it, these four sort of globular proteins kind of binding together, and each one of them um, has a binding pocket where oxygen can bind. And each time one of them binds oxygen, it makes it easier. It opens the structure a little bit so that oxygen can be bound by the other hemoglobin. And so this is the normal way. And so oxygenated and deoxygenated are just indicated this way. But the mutant has this sort of sticking out part of it that causes not the correct binding, but causes a fiber to form instead. And it's literally just this one amino acid difference that causes the binding to shift, right? And so when the molecule has no oxygen bound to it, it will form these weird fibers. You can see how they fit together. And so this causes the deformation of the blood vessels, and it causes pain, tissue damage, and lack of energy, exhaustion, and so on. And that's the tissue damage is because of the deformed cells, but the ability to carry oxygen is what causes the problems with fatigue and sometimes even heart effects and so on. And then this is just a picture. This is a normal red blood cell. It looks like a donut, except there's no hole in the donut, just a little dip. And there are, in a person who's heterozygous, some of the cells don't look 100% normal because they have some normal and some abnormal hemoglobin inside. But a cell that has all abnormal hemoglobin has this sickle shape because the proteins don't form these nice globular structures or globular shapes where they bind, and that's what leads to the sort of dip in the middle. And when the protein binds in this sort of fibrous way, it kind of deforms the proteins. It deforms the cells, so they have this unusual shape. It's really interesting. You might want to look up online because some years back they started working on genetic gene therapy for sickle cell anemia, and some people are now being treated for this with genetic engineering. One of the things I talk about in my genetic engineering class. Okay. So that's one example of a very serious missense mutation. There's also what are called, nonsense mutation. Nonsense is like no sense, and that is because a codon for an amino acid is altered into a stop codon. Okay, and again, this is where remembering the stop codons can be very valuable. It can save you time. So these will cause a truncation in the protein. So, and again, if you have this whole protein sequence and you've got a nonsense mutation right at the tip, the C terminus, and so the protein is just a little bit shorter than normal, that may not have a very strong effect. It might have just the mildest of symptoms or phenotype. But if it happens, elsewhere in the gene, it usually will eliminate the gene function, because a complete protein, sometimes only the first few amino acids of a protein are made. There's no way that's going to be wild type. There's no way that's going to be a healthy, normal protein. Okay, so this is the example. And this is, again, our coding strand and template strand. The change is here. So instead of a GC pair here, there's a TA pair. And what that means is the GAG, which should have encoded GAG glutamate, is now a UAG, which is a stop codon, right? And so there's nothing to, when you're writing out your amino acid structure, you do methionine, proline, glycine, and then sometimes students want to do something like write stop or something like that, but instead just put the C, because the C means that's the end of the protein. So when you write it like this, you're communicating that's the stop codon. Because when we're writing out the normal gene sequence, we never go, okay, this is the stop codon, and write stop or anything. That's the end of the amino acid sequence, and then we put the C terminus there. Okay, that's just a conventional way of describing it. Okay, so we'll also talk about regulatory mutations, and we'll talk about them later on when we talk about gene regulation in prokaryotes. Maybe if we have time, we'll look at some eukaryotic stuff, but most likely we'll stick with the prokaryotes. But just think in general, if you were to delete a promoter sequence of a gene, your RNA polymerase can't bind, right? So you're not going to get any transcription, and therefore you're not going to get any protein. If you hugely change the promoter sequence, remember the -10 sequence and the -35 sequence, the Tata box at -10 and the other sequence at -35, these are absolutely required for RNA polymerase binding. So if you make a huge change in one or the other of these, you're going to basically eliminate gene function. You can make sometimes minor changes in the promoter sequence that have mild reductions in transcription efficiency or something. We're not really going to be talking about these. You may cover something like this in other classes, but in our case, in our class, the regulatory mutations that we're going to talk about are the ones that have a huge effect. So either RNA polymerase can bind or it can't bind. And you'll see what some of the others are when we get to the lac operon. But we're not going to be looking at subtle, what happens if you change one nucleotide in the Tata box or something like that, because probably not very much will happen, right? Okay, and then just in terms of translation, the in vitro work in a test tube doesn't necessarily use the normal signals, like starting at a start codon and that sort of thing. The enzymes in the system used don't require that. So you basically just start at one end of the RNA and then just go to the end of it for the purposes of this class. So if I tell you this is done in vitro, then you just take the DNA and decide what the RNA is, and then just tell me what the protein will be. And this is the kind of technique that was used in the last lecture, figuring out how the genetic code worked. And then in vivo translation means inside a cell, right? So you need the proper ribosome binding sites, which we'll talk about in the next lecture, and they always start at the start codon. So it makes a difference which of these systems I'm asking you about. Like, the answer will differ, and so it's important to ask. If the question doesn't say in vivo and in vitro, then please ask and I will clarify. Okay, and then just one final thing is that all the mutations that we have been talking about are what we call loss of function. That means the mutations all are they producing either a slightly or a very defective protein product, or very little or no protein product. So this is loss of function. Loss meaning that the protein isn't there, or it's not doing its job, or it's not doing its job very well at all. We may talk about gain of function mutations later when we're talking about some of the some other kinds of gene regulation. So I cover this a lot in some other classes. We may get to it, but just think of these so far as all loss of function mutations. So translation requires your mature messenger RNA, a set of tRNAs, one or more that carries each of the amino acids, tRNA synthetases, these are enzymes for each tRNA. They link the correct amino acid to the correct tRNA. And there's sequence that's required for that. We will just talk very generally about this. The ribosome, which is RNA plus proteins, it's little molecular machines that actually provide the scaffold for peptide synthesis, and they do the reaction. They produce the peptide bonds and other proteins and factors. And this is what we're going to talk about in the next class.